her and the other group of folks. We had 6 people working on various questions, related questions last semester, and 3 of them are going to be talking later this week or next week. And I'm going to stop talking now. I'm going to Kennedy tell you about her project. She's going to do a good job. Yep.
All right, everyone. My name is Kennedy Snow, and I'm really excited to be sharing my research with you today. A little bit about me before I get started is I'll be graduating in about a month here with a bachelor's in biology, environmental science, and nutrition. Outside of academics, I love to cook, read, draw, spend time in nature. And for the last 5 years, I've worked as a nurse assistant, and the last 3 of those years, I've done travel nursing, where I go to different facilities in the Madison area and pick up shifts when they are short-staffed. And that's actually one of the big things that inspired my interest in this project. That work gives me a real-world, day-to-day look at how things like infections and antibiotic resistance impact people on a day-to-day basis. And I really love thinking about the world that we live in and asking big questions. And I think genetics, the basis of my project, is a great way to do that. And also connecting science with my faith. I believe genetics is a beautiful way to explore that. Genetics is truly one of the languages and arts of God, and it was a privilege to learn about it on a deeper level. So today I'll be walking you through my research called Unlocking Yeast Drug Resistance, the roles of SSC1, PDR1, and quorum sensing molecules. There is a mystery right now. Somehow the culprit, pathogenic yeast, continues to get away with causing havoc because it's outsmarting our best drugs. We need to better understand the mechanisms that are allowing it to do this so we can create better antibiotics for those affected. So as I said, there is a growing crisis. Hospitals and healthcare centers around the world are facing antibiotic-resistant infections that refuse to go away. Yeast infections in particular are a difficult problem because they are eukaryotic, like our own cells, so they share a lot of similar components to us. It's difficult to create drugs that will harm the yeast without also harming our own cells in the process. Yeast, particularly species within this genus Candida, are a fungi that naturally inhabit our skin and mucous membranes, and they usually don't cause us harm, but it's when they overgrow that various infections can arise, and people with weakened immune systems are particularly susceptible. Vaginal candidiasis, commonly known as a yeast infection, is the most prevalent fungal infection affecting women. And then Candida can also overgrow in your mouth or throat. You may have heard this as oral thrush. As a nurse assistant, the most common infection I see is when it overgrows in their skin folds. So they have discomfort in areas like their groin, chest, stomach, and like underarm areas. But the most concerning type is invasive candidemia, and this is where it enters the bloodstream. It's most commonly affecting hospitalized patients, and it is associated with mortality rates between 30 and 60%, which is really high. The CDC estimates that candidemia is the most common bloodstream infection in the United States, and according to a review led by Felix von Goman, there are 700,000 cases worldwide each year, and that's a statistic from 2021. A new article was posted March 22nd, so just a few weeks ago, that the CDC has now labeled this as an urgent threat because cases have been tripling every year for the last few years. This is a great visual showing why candidemia, the bloodstream infection, affects hospitalized patients mostly, and basically it's because there's an increase in avenues for the pathogen to enter their body. So you may be undergoing an invasive procedure where it could enter through your open body cavities. There's different machines like blood glucose monitors or ventilation systems where it could transfer from one patient to another since they're often shared. lingering in the room and staying because of improper disinfection. And it could also enter through personal devices. I often see it entering through like catheters, PICC lines, or prosthetics. And then of course we have the antibiotic use itself. This is where the antibiotics are prescribed. So this is the start of the yeast being exposed to the antibiotics they will soon be able to resist. So the development of multi-drug resistant strain is increasing the concern we have of this pathogen. It's becoming a serious global threat as it continues to outsmart our drugs. This is where my research comes in. I wanted to investigate how the yeast cells are able to survive despite being in the presence of these drugs, and I wanted to understand the mechanisms involved in hopes that we could contribute to the knowledge we have to make better antibiotics. So let's get into how the yeast are able to continue to outsmart us. Pleiotropic or multi-drug resistance, also known as PDR, is the system that the yeast use to get the drugs out of them. The key idea here is that yeast can literally pump out the drugs from their cells using these membrane pumps on the membrane. There are many genes involved, but the one I chose to focus on is PDR5. This is a gene that when activated and then transcribed and translated, makes these proteins on the membrane. So I just said these words, transcription and translation. Let's do a little quick review into what those terms mean. Transcription is the first step in gene expression. It's when the gene's DNA is copied, and it's copied by this enzyme called RNA polymerase, which is this red circle here. When we think of DNA, we often think of this double helix structure, and in this process, that structure is unwound, and then corresponding bases are brought in to create this red mRNA strand. This red strand can leave the nucleus and go to the cytoplasm to be translated into a protein. So after transcription comes translation. This is the process where the mRNA, that red strand that was just created, can now be read by the ribosome, which is shown in this green circle here. The mRNA is read in groups of 3, and these 3 bases are called a codon. Each codon codes for a specific amino acid. So in this picture, AUG is coding for methionine, AAG for lysine, and when these amino acids are linked together and then later folded according to their properties, that's what forms this final protein structure. And in this case, what makes those membrane pumps. So now that we know how the PDR5 gene is expressed, let's learn how it is turned on. It doesn't just turn on by itself, it is turned on by this PDR1. PDR1 is an activator, which means it binds to a specific region of the DNA in this case, of the PDR5 gene. And it plays a crucial role in the recruitment of that RNA polymerase so the gene can be transcribed. But PDR1 also doesn't just turn on by itself. The activator needs to be activated. Most PDR1 articles indicate that it's maybe activated by drugs, but this is poorly understood, so my research looked at the protein-protein interaction between PDR1 and SSZ1 for its activation. So what is SSZ1? This protein is normally attached to the ribosome, that protein that is involved in translation, but it's suggested in some literature that under stress, like when there's a drug present, or under nutrient stress, that it will detach from the ribosome. Once it's free, SSZ1 can move into the nucleus to bind to PDR1. And that binding changes PDR1's shape, activating it and allowing it to thus turn on PDR5. So on my title slide, I mentioned quorum sensing. How is this related to this system? So quorum sensing is a form of cell-to-cell chemical communication, and it's basically where microorganisms like yeast can talk to one another using little signaling molecules. When enough of these signaling molecules build up, it can trigger a coordinated response across the entire yeast population, and it could lead to changes in gene expression, stopping cell growth, or activating these drug resistance pathways. One interesting example happens under nutrient stress, specifically when nitrogen is limited. In this case, the cell takes nitrogen from amino acids to use for the biosynthesis of other things it needs in the cell. And in that process, fusel alcohols are made. The cell doesn't use these fusel alcohols, so they're released into the environment as waste. Specifically, 3 of the quorum sensing molecules I looked at use this pathway. Phenylalanine is turned into phenylethanol, tyrosine into tyrosol, and tryptophan into tryptophol. But what's fascinating is that these molecules are not only waste products, but they can also facilitate that communication between cells. Those neighboring cells can detect these molecules, but the effect that they have on them is still poorly defined. Other than those 3 quorum sensing molecules, I also studied farnesol and neurolidol, and they come from a different metabolic pathway, specifically sterol biosynthesis, where it makes certain sterols, some being the fungal equivalent to our cholesterol. Basically, in this pathway, farnesyl pyrophosphate, or FPP, shown in these red boxes, is the essential intermediate. But when too much of that is made, the cell diverts it into forming these farnesyl and neurolidol quorum sensing molecules. And just like the fusel alcohols I just talked about, farnesyl and neurolidol are not used by the cell, so they are secreted as waste products. But they too have secondary functions. From previous literature, we know that farnesyl is a signal of protein population growth, or just of population growth. It's important for fungal virulence, and we also know that it activates PDR1 in at least 1 species of Candida. Neurolidol is mostly found in biofilms, so the amount of it increases as the formation of these happens. And both farnesyl and neurolidol can cause changes in cell morphology, but it's still not understood how they are secreted out of the cell or how they are detected by neighboring cells. Their roles as quorum sensing molecules are still being unraveled. So that was a lot of information. Let's just look at this simplified image to kind of review everything that I just went over. So we have this PDR system. This is the drug resistance system that the yeast is using. We have SSZ1 that has detached from the ribosome, has entered the nucleus, and is now bound to PDR1, which is activating it. Now that PDR1 is activated, it can bind to the PDR5 gene. And so now PDR5 can be transcribed and translated to make these plasma membrane drug pumps that will be pumping out the antibiotics and maybe also the quorum sensing molecules. For what I specifically investigated, I conducted 2 main experiments. First, I looked at that protein SSZ1. I wanted to know what amino acids of SSZ1 are important for its activation of PDR1, and also if we can affect drug resistance by mutating specific parts of it. Then for my second experiment, I looked at quorum sensing molecules. I wanted to know the effect certain ones had on yeast cell growth, and if any can directly cause early growth arrest. And then to connect these 2 ideas, I asked, do quorum sensing molecules somehow trigger or use that PDR system? So for the first experiment, I started by using transformation to insert plasma DNA into the yeast cells. Each plasmid had a gene that encoded for a different version of SSC1, which is shown here on the screen. The amino acids, so if we look at this picture, the 295th is in purple, and then the two that I tested, 239 and 238, are in green and blue. And I chose these amino acids because of the knowledge we have from previous literature. That literature told us that this 295th one has been deemed important for the folding of SSC1 and thus its activation and binding to PDR1. And we can see that because of how this protein twists and turns and folds, these three amino acids are near each other and are thus interacting with one another. So I wanted to see if the 239 and 238 amino acids are important, like this purple one. So how were these amino acids mutated? This is a chart of the 20 amino acids that we need in our bodies. The amino acids are all similar in structure. They all have this amino group, this carboxyl group, and this hydrogen. But where they differ is this R side chain. And that's really important because that's what gives the amino acid its unique characteristics. Some are positively or negatively charged, some are polar and uncharged, and some are hydrophobic. And these qualities are important for how the protein folds. We saw in the previous picture that there's lots of twists and turns to the protein, and that comes from the properties of these amino acids. So for my experiment, I looked at just 4 of the 20 amino acids, and they're highlighted in these red squares. And on the left are the 3 mutations that I did. And I first want you to understand the notation of these mutations. So the amino acid that is mutated is the number in the middle. The letter on the left corresponds to the original amino acid that's in wild type SSC1. And then the letter on the right is the amino acid that I mutated it to. And yeah, so let's zoom in on those 4 that I looked at. We again see that they all have the same basic structure. They all have this HO at the top. They all have this double bonded oxygen, and they all have this NH2. But where they differ is this bottom R side chain. For the first mutation, I went from an E to an S, which is changing a glutamic acid to a serine. We're going from a negatively charged amino acid to a polar uncharged one. This matters because glutamic acid often forms ionic interactions or salt bridges due to its negative charge. So replacing it with the serine removes that negative charge, and it allows us to test whether those electrostatic interactions are important for the binding to and activation of PDR1. For the second mutation, we're going from a Y to an S, which is changing the tyrosine to a serine. Everything is the same here, except we're removing this aromatic ring of carbons and hydrogen. These aromatic rings are capable of forming hydrophobic interactions, which again are often important for SSC1 and PDR1 to interact. So it allows us to investigate if that is crucial for turning on PDR1. And then finally, for the third mutation, we're going from a Y to an F, which is a tyrosine to a phenylalanine. This is similar to that second amino acid, but now we're keeping this ring and we're just getting rid of the OH group. The hydroxyl group can form hydrogen bonds, so by removing it in this case, we're looking if that is important for interacting with PDR1. So to continue my methods, to investigate the effect these mutations had on drug resistance, we measured cell growth in the presence of the antibiotic cyclohexamide. Cells with the specific plasmids were grown in liquid media and then treated with the antibiotic. And then once grow for another 24 hours. I then used a spectrophotometer to measure the cell density or absorbance, both before and after the 24-hour period. This is just a quick reminder of how the spectrophotometer works. The cell sample is here. Light passes through the machine and goes through our liquid media sample. And the more cells that are in the sample, the less light that we'll be able to get through. So that tells us about the cell density and how many cells we're able to survive. For my second experiment, yeast cells that were actively growing were treated with one of the 5 quorum-sensing molecules I've talked about earlier, 2-phenethanol, tyrosol, tryptophan, farnesol, or neurolidol. Again, to see how they grew, I used a spectrophotometer to measure their cell growth, both before the quorum-sensing molecules were added and then 2 hours afterwards. And then for both of my experiments, I did statistical testing to see if any of my results were significant. I used the Wilcoxon rank sum test to do this. This is a non-parametric test that compares 2 groups. To kind of talk about what that means, a non-parametric test means I don't assume anything about the distribution of my data. In the field of molecular biology, we use non-parametric tests as a rule. Doing just 3 replicates is the norm, so with such few replicates, it's difficult to be confident about our data being normally distributed. This, however, is not an easy way out for stats because the bar to see a significant difference is actually higher in a non-parametric test. The data is messy, so the test makes less assumptions. This means there actually needs to be a bigger difference between the groups to actually have a statistically significant p-value. This is a conservative way of doing stats and actually makes you more confident about your results. The paired groups part of the definition just means that you're comparing 2 sets of measurements that are somehow meaningfully connected to one another. So like a before and after measurement of a treatment. And then before I get into my results, I just wanted to quickly reiterate my investigation questions. I wanted to see if certain amino acids of SSC1 were important for the activation of PDR1, and if we can affect drug resistance in any way by mutating specific parts of SSC1. I then wanted to see if certain quorum-sensing molecules affected cell growth or caused early growth arrest, and then to connect these 2 ideas, if quorum-sensing molecules somehow trigger or use the PDR system. So these are my results for experiment 1. To orient ourselves to this graph, we have my absorbance levels taken with the spectrophotometer on the y-axis, and then my mutations on the x-axis. Mutations 239 on either side and 238 in the middle. And then we have the levels taken without the antibiotic in blue as our control, and those treated with cyclohexamide in orange. After statistical analysis, we found that there was no significant difference between any of these mutations. However, for this experiment, I did not have an ideal control. In retrospect, we should have had our control be a wild-type version of SSC1 instead of also having the mutation, just not being in the presence of the cyclohexamide. So this makes it difficult to make strong or clear conclusions. So therefore, I haven't for this experiment really answered that first question yet, but further investigation and redoing it with a better control would help clarify it. Another future experiment would be using different concentrations of cyclohexamide. There's only so much that the cell can handle and pump out, so maybe I was overwhelming the cell with the amount of antibiotic. So I would be interested in redoing it with a smaller concentration. These are my results for the second experiment. The graph shows the differences of yeast cell growth in the presence of the different quorum sensing molecules compared to my control, which did not have any quorum sensing molecules. The y-axis again shows my absorbance levels taken with the spectrophotometer, and my x-axis of my control and my 5 different quorum sensing molecules. And then after statistical analysis for this one, I did see a significant amount of growth arrest in 3 of my quorum sensing molecules, 2-phenylethanol, farnesol, and geraldol, but I did not see a significant difference with tyrosol and tryptophan. For this experiment, I learned what we were trying to ask. I believe what my data is telling us that some quorum sensing molecules are causing growth arrest and some aren't. And this makes sense. There's different quorum sensing molecules for a reason, so maybe tyrosol and tryptophan are just telling the cell to do something else instead of cause early growth arrest. For future experiments, I would like to see how the yeast cells would respond to the quorum sensing molecules without having SSC1. My control still had the SSC1, it just didn't have any quorum sensing molecules. And this would kind of get at my third question. If SSC1 was needed to see these results, that could suggest that the quorum sensing molecules are using the PDR system to get out of the cell. So overall, my research helps us better understand the mechanisms involved in yeast cell antibiotics. Like I said at the beginning, yeast cells are eukaryotic like our own, so it's difficult to develop drugs that will target them without harming us. But we do not have a PDR system, nor do we have quorum sensing molecules. So if we can understand these yeast-specific mechanisms, we can create antibiotics that will target them and not hurt us. And of course, I would like to give out a few thank yous before I finish. I want to give the biggest thank you and shout out to Professor Pete Kuhn for being my mentor. He truly guided me through every step of this process, and he dedicated a lot of time coming to our early morning meetings every week to really help me conceptualize these big ideas and edit my project. I really couldn't have done it without him, so thank you. I also want to thank Jake Griffin and Tim Kuhman for their help in seminar, for providing resources that helped me become a better storyteller of science. I want to thank Bryn and Cassidy for being my peer review buddies. They helped me stay on track in completing this big project, and I appreciate their insights in editing it. And then lastly, I want to thank my beautiful parents and my love Mo for being my motivators and encouragers when this project felt really overwhelming at times. They often reminded me of the light at the end of the tunnel that I am now in. And then, of course, I want to thank everyone here right now for your attention, and I would love and be happy to take any questions you might have.
All right, thank you so much, Kennedy. Questions, comments, discussion from Bio 480 students?
I was just curious if there was like a reason or rhyme to why you use cyclohexamide, and from your past literature searches, do you know of any other antibiotics that might have shown a more significant result?
Yeah, we used cyclohexamide first because one of the literature papers we read used that one, so we also used that one. Cyclohexamide targets the translation mechanisms, so it's like a good way of seeing PDR1 interactions, because shutting down the making of those clumps. But cyclohexamide isn't used anymore it's not prescribed anymore because we too have translation mechanisms, so it was causing bad side effects. So yeah, I guess I would be interested in seeing if my results were the same with antibiotics that are actually used in the real life.
Having worked in the healthcare system now for many years, is there any practices you've noticed that could get better to kind of avoid this drug resistance that we're obviously is increasing now?
Yeah, in my own work, I really only see one antibiotic ever used for yeast infections. It's called nystatin. It's like this powder, kind of like a baby powder that you just put in like the folds of their skin where they have a yeast infection. And honestly, like, it's just used so much. I've never seen it really improve anyone's condition. It's just something that they put on every single night. So again, I would be interested in seeing if we could use that somehow in my research and if that affected it. And maybe if there's like a better one out there. But yeah, it doesn't really seem to be that useful.
Once it hits the bloodstream, do you know how this is tested for in hospital settings as far as patients?
I honestly haven't looked into that, but I would assume like a blood test of sorts could like detect the fungi in your blood as it's just like moving around.
All right. Other questions?
So you had a couple of future research topics, like having a wild control as opposed to a modified control, things like that. And to go along with what Red had asked with the particular antibiotic that you use, would you try another experiment, let's say with nystatin, to see if it even does anything to these? Like obviously we have some form of medical research or else they wouldn't be using it, but maybe that was from yeast infections that happened in the 50s and we just haven't updated that because it worked at one point and now we have so many, as you said, there's a UN.
Yeah, I would be definitely interested in testing this again with nystatin specifically, and like using other strains of Candida as well. In my experiment, we used baker's yeast just because it's the non-pathogenic to me as a researcher. But I would be interested not only using different antibiotics, but also how those antibiotics impact real-world pathogenic yeast, because that too could lead to different results.
How did you get into sorry?
How did I what?
How did you obtain the mutants? Like,
Keith has a little collection of mutants that are like, some of them are like a decade old, some of them are like 50 years old, but from his like past graduate work. So my work was mostly like stressing the yeast to uptake the plasmids that were already created.
did you verify?
In what way?
Like, did you look for it? Says SSC1, right? That you made. Did you check that you were expressing SSC1? Did you do a Western blot?
We just knew that the certain cells had taken in the plasmid DNA because when we put it on the plate, the only way they'd be able to grow on that specific medium is if they had the plasmid. So we just took those cells that were able to grow and then treated them with those specific ones.
It was only a really small amount of that protein.
Maybe.
That might affect.
Yeah. We also thought about that too, like how some of them are kind of old, so that might like affect how much they were able to grow.
Can you put up one of your absorbance bar graphs? Yeah, it's perfect. So, is there a linear relationship in between absorbance and the population size of cells in your tubes? So, if we see a 50% reduction in absorbance here, let's say for your treatments there in the middle, does that equate to necessarily to a 50% reduction in growth, or is there a nonlinear relationship between absorbance and population size? How do we kind of think about population sizes or growth if we're measuring absorbance?
Yeah, so like I think what you're getting at is like the absorbance is telling me about cell density.
Right.
So the amount that they decreased after being exposed to the quorum sensing molecules would tell me how many like died or like stopped growing after the treatment.
Yeah, so is that a linear relationship? Like if you plotted number of cells in your tube versus the absorbance value, is it a straight line or is it curved in some way?
I feel like it would, like my speculation is that it would be kind of like a J shape,
Okay.
and where like growth arrest is gonna stop, but eventually it's gonna like plateau and level out, which is something that different groups are looking at right now. Is like how is that growth arrest actually like quantified? Because I didn't necessarily look at like specific numbers. I just kind of looked at general absorbance. So some people are looking at specific growth charts of it.
I guess, continuing off that, how do you know in this data that the cells you're measuring are alive or dead, when the dead cells still cloud the absorbance rate?
That is a good question. To be honest, I don't know. Yeah, I don't know. That's a good question. I feel like we did something in our methods to account for that, but I don't really know.
They might be dying, this doesn't differentiate those, but these are all actively growing cultures. So we're really looking at how much they increased, and some increased more than others. But this approach is blind to whether they're on their way out or, not very functional anymore and dying or normal cells, just looking at total population. But this is taken in the context of without the quorum sensing molecules, it's logarithm growth. And so we're not starting out with lots of cells and then seeing how many survive. Really, we're starting out with a low number of cells, letting them grow and seeing how much less growth happens. And so it's be a really small percentage of dead cells unless we hit toxic levels of these things. And that would be another experiment that is. Right, that's one question we didn't ask, but could ask is, are some of these cells, is growth less because they're safe? And we didn't look at that, just seeing less growth.
That was really great, thank you.
Thank you.
I think I remember you saying this, that farnesol is a particularly virulent form, and is it virulent because it increases PDZ or activation of PDZ, or is there some other reason?
The quorum sensing molecule I said affects virulence is farnesol, and that comes from just other literature that has looked at farnesol, and it just alluded to it being important for virulence, but I didn't really necessarily know how or why.
Okay. And then, for the flow spectrometry, why 600 nanometers? I mean, is there a particular reason related to cells that spectrum?
I believe in like molecular chemistry that's kind of like the yellowish wavelength, and so these cells that are kind of yellowish, so that's just kind of also like the standard, like we just kind of do that one often, but I think it has to do with color.
Yeah.
Other questions or discussion points? Okay, we have lots of time.
Yeah.
Can you go back to your methods to slide?
Yeah, so when you added the quantum sensing molecule, how did you decipher, did you test each of them, or did you do, I mean, made clear that you did, it was stated, then how did you, I guess, was there a certain method of reason of why you went about which one goes first, which goes second?
They were kind of all done at the same time.
Okay.
So they all had their own little tubes and then they were just inserted. I had 3 different replicants for each of the 5 corpses. So they were done kind of the same time.
Okay.
Yeah.
What is the generation rate for a non-treated yeast, as an example? Like, how fast does the population grow? Like, you said it was logarithmic growth, but like on what time scale? Because it looks like, your experiment is after 2 hours, how much the population, or theoretically the density of those cells, if nothing else, increased.
That's a good question. For mine, we let them grow, I believe over 48 hours was our time scale. But there's someone this semester who's playing with that to see like, is that too much, is that too little? But it's like they're on a fast time scale, like it's not, you don't have to wait that long for them to grow. And if you wait too long, then there's too many cells to really do it. So yeah, like 2 days.
But how long is like how long does it take for, you start with a batch of cells, how long does it take for that batch of cells to generate a generation of cells? Because that's on the order of minutes, seconds, milliseconds. How long does it take?
I honestly don't know the differentiation, if it's like seconds, hours, but I just know that it's not that long. Like we were able to see results for this experiment after just 2 hours, so it's pretty quick. Yeah.
About an hour.
About an hour for
For generation time.
Okay.
So we're letting it, the goal is to let them double twice a year.
I missed this the first time when you showed the slide. You took the spectrometer reading like immediately after giving the form sensing and then waited 2 hours. Right? So now if you go forward to your figure of your results, right? This one, yeah. Now the absorbance on the y-axis, is this the absorbance after 2 hours?
Yeah, for the 5 fluorescency molecules, that's after.
So why do you take a reading immediately and then after 2 hours, but not? I guess the question is why didn't you use the percent change from the first reading to the second reading? What was the point of the first reading?
That was more like our control, like before the fluorescency molecules had a chance to have an effect on them.
Did you see much difference between the treatments at the immediate spectrometer reading?
No, that was like, the controls were generally the same. It was, the differences came after the 2 hours.
Yeah.
They had time to have an effect.
I don't know if this is too much, so just tell me if it's too much. You don't need to answer this part. So why would yeast make cholesterol when it was stressed?
Under stress.
Yeah, because corn signaling liver. Or not necessarily.
Not necessarily. For the first 3 that I talked about, so in this situation with like farnesol, tryptophan, tyrosol, this is kind of a stress response when they don't have nitrogen.
Yeah.
But this one isn't necessarily a stress response. Like they're already making this to make these sterols. It's when like they don't have, when they're over making it, that they start making neuronal and farnesol. So I wouldn't necessarily call that like a stress response.
Okay, I just didn't know if like they were stressed alone.
No, I think they're just like waste products from like oversaturation.
No, I meant the end product, like this is like end product.
Yeah, I think those are just used in a membrane as
Okay, so usually
sterols.
in cell.
Yeah, yeah.
All right, let's let Kennedy off the hook. Thank you so much, Kennedy.